we've got Alexandr Ilyaskin. Now, Alexander is a Privatdozent in the Department of Cellular and Molecular Physiology in the Friedrich Alexander Universität of Erlangen-Nuremberg. He has extensive research experiments in renal physiology and also the complicated interplay of proteins involved in blood volume regulation. And today, we're very happy that he's here with us, that he will give us a talk about his recent work studying the epithelial sodium channel, ENaC. Alexander, thank you very much for being here, and the floor is yours.
So ENaC is responsible for sodium reabsorption from the urine back to circulation, and thereby it controls how much sodium is excreted with the urine. So if ENaC activity is too high due to hyperaldosteronism or as a result of a gain-of-function mutation by pseudo-hyperaldosteronism, too much sodium is reabsorbed. And this sodium retention causes hypertension. Importantly, very many drugs to treat hypertension ultimately inhibit ENaC activity, either indirectly by inhibiting renin-angiotensin-aldosterone system, or more directly by inhibiting mineralocorticoid receptor like spironolactone, or blocking the channel itself directly, like amiloride. On the contrary, if ENaC activity is too low, sodium is not reabsorbed enough and excreted with the urine, which leads to hypotension. And these patients may probably benefit from ENaC activators. ENaC plays also a very important role in lung. Here, ENaC, luminal membrane, represents the major step for sodium absorption from lumen into ciliated airway epithelial cells. This sodium influx, together with chloride efflux mediated by CFTR, controls the volume of airway surface liquid. This process is disturbed in CF, in cystic fibrosis patients, and these patients probably may benefit from inhibiting ENaC, so by using ENaC inhibitors. ENaC is also expressed in alveolar epithelial cells, where it is responsible, contributes significantly to sodium reabsorption from alveolar lumen. ENaC activation here would promote removal of sodium and water and help patients with pulmonary edema and respiratory distress syndrome to improve fluid clearance from alveoli. So I hope I can convince you that ENaC is very important and established drug target, and both ENaC activators and ENaC inhibitors can be used to restore normal ENaC function in lung and also in the kidney. And this justifies research efforts to study molecular mechanisms of ENaC regulation. It was not until recently that the structure of ENaC was resolved by means of cryo-electron microscopy. So this is how this channel looks like. So channel consists of alpha, beta, and gamma subunits. And now having this structural information, we're able to perform computer simulations and combine them with experimental approaches to study molecular mechanisms of ENaC function and regulation, to elucidate pathophysiological mechanisms of diseases associated with disturbed ENaC function, and probably to identify novel pharmacological and endogenous ENaC modulators. So in general, this structural information opens new exciting horizons to study ENaC on a molecular level. We call this combined approach structure-based function analysis of ENaC. And in the rest of my talk, I will provide examples of studies where we use this combined approach to answer questions which we are interested in. So first, we use this combined approach to study how this channel is activated by proteolysis. Proteolytic cleavage of ENaC is its unique feature, its unique stimulatory mechanism. Proteases cleave alpha and gamma ENaC at specific sites, which leads to the release of autoinhibitory peptides, shown here in red and in blue. This release of inhibitory peptides activates the channel. Alpha ENaC is cleaved by furin or furin-like convertases at these two specific sites. This probably occurs intracellularly during channel maturation and trafficking to the plasma membrane. There is also one furin cleavage site known in gamma subunit. But when channel is cleaved only here, it doesn't reach its maximal activity. Γ-ENaC should be cleaved at the second site, at the distal site. This is so-called final proteolytic activation step. And when it's cleaved twice in gamma inhibitory peptide is released and channel reach its maximal activity. In vitro, many proteases can cleave γ-ENaC at this site. For example, a prototypical protease chymotrypsin. And recently, we have identified protease, transmembrane serine protease 2, TMPRSS 2, which does this job in vivo in kidney. And we have recently published this. With the help of xenopus oocyte expression system, we studied molecular mechanisms, how TMPRSS 2 activates ENaC proteolytically. So xenopus oocytes can be isolated from female frogs. Oocytes can be injected with cRNA encoding all three ENaC subunits, alpha, beta, and gamma. And after 2 days of incubation, protein is formed and heterotrimeric channel is formed at the cell surface of an oocyte. In oocyte expression system, alpha subunit is fully cleaved, but gamma subunit is only partially cleaved. Therefore, these channels are only partially active. And we can measure this ENaC-mediated currents with the help of 2-electrode voltage coulomb technique, and we have obtained these results. So here you can see amiloride-sensitive currents in oocytes expressing alpha, beta, gamma, ENaC. And these currents are pretty small. If we co-express ENaC together with TMPRSS 2, TMPRSS 2 protein is formed, active protease is formed at the cell surface, which leads to cleavage of gamma subunit and full stimulation of ENaC activity. And indeed, in oocytes co-expressing ENaC together with TMPRSS 2, pretty large currents can be observed. So TMPRSS 2 stimulated ENaC more than 4-fold in these oocytes. After that, we studied molecular mechanism, how TMPRSS 2 stimulates channel, and we were able to demonstrate that this occurs due to the release of gamma inhibitory peptide from gamma subunit. So we analyzed the structure of gamma subunit, particularly the binding site of gamma inhibitory peptide. We identified 2 amino acid residues, one in gamma inhibitory peptide and the second in the binding site, which were close enough to each other to form a disulfide bond when they are substituted with cysteine residues. So we introduced novel disulfide bond between inhibitory peptide and the binding site, and we wanted to block gamma peptide release and activation of the channel, even when it is cleaved by TMPRSS 2. And we were lucky to confirm these predictions experimentally. So indeed, double mutant with this novel disulfide bond cannot be stimulated by co-expression with TMPRSS 2, unless this disulfide bond was broken by incubation of oocytes with a reducing agent, DTT. In parallel control experiments using wild-type channel, we observed stimulation of ENaC currents by TMPRSS 2 as usual, and DTT produced no significant effect on the currents, demonstrating that this effect of DTT was specific. So to conclude, with the help of structural analysis, molecular linkage approach, and 2-electrode voltage measurements, we were able to elucidate molecular mechanism of TMPRSS 2 mediated ENaC stimulation and demonstrate that it was due to gamma peptide release from gamma subunit. Importantly, proteolytic ENaC activation can be reversed. Synthetic peptides, which have same sequence as endogenous peptides, can be synthesized and applied in extracellular solution to inhibit the channel. So here you can see an example trace demonstrating this phenomenon. So ENaC can be stimulated proteolytically by applying a prototypical protease chymotrypsin, which removes endogenous peptide from gamma subunit. And after that, we apply synthetic peptide mimicking endogenous sequence, which inhibits the channel. So the currents reach initial level. We analyzed the binding site of inhibitory peptide within gamma subunit and identified critical amino acid residues which stabilize this binding within the binding site. Of particular importance was tryptophan residue 229. We mutated this tryptophan to alanine and observed following results. So baseline currents were pretty much bigger at the beginning of the measurement, even in the absence of protease at the cells in the bath solution. This was probably due to the fact that endogenous peptide, which is still there, can no longer bind to this site and produce inhibitory effect. Consistent with this interpretation, application of chymotrypsin in the bath solution produced no further stimulatory effect. And importantly, application of synthetic peptides also produced no inhibition, meaning that both endogenous peptide and synthetic peptide can no longer bind to its binding site and produce inhibitory effect. This information, this characterization of the binding site of inhibitory peptide is of particular importance from pharmacological perspective, because it demonstrates that theoretically it is possible to design, to develop novel ENaC inhibitors which bind to this site. Probably this could be peptidomimetic inhibitors which can bind to this site. And we can design novel antihypertensive drugs to treat hypertension. We would like to pursue this aim in our further research. And also, as I mentioned in introduction, not only ENaC inhibitors, but also ENaC activators are of importance. What do we know about ligand-mediated ENaC stimulation? Only one molecule is known to date to stimulate ENaC. This is the small molecule ENaC activator, S3969. It has been developed in 2008. This is also a peptidomimetic compound consisting of indole ring, found also in tryptophan residues, and leucine serine dipeptide-like moiety. In this first publication, it has been demonstrated that human alpha-beta-gamma-ENaC, but not mouse alpha-beta-gamma-ENaC, can be inhibited by this compound. And also it was demonstrated that extracellular loop of beta subunit is responsible for this stimulatory effect. We were able to reproduce these findings. We were able to demonstrate that human, but not mouse, ENaC is stimulated, and that this portion of beta subunit is really important. So here we co-expressed mouse alpha and gamma subunits together with a chimeric beta subunit in which extracellular loop was replaced by human sequence. And here you can see that this chimeric channel can be stimulated as effectively as human autolog. Next, we wanted to identify the binding site of this compound because it was known. It was not known where this compound binds to beta subunit. And we generated a series of different chimeric beta subunits to narrow down the region within this extracellular loop. And we were able to identify the central portion as the most critical region for this stimulation. After that, we looked at the structure to find a putative binding site within beta subunit which corresponds to the identified sequence. And we were lucky enough to find a cleft formed by parts of thumb domain, shown here in green, and parts of beta ball domain, shown here in red. And with the help of molecular docking approach, we demonstrated that S3969 molecule can bind to this cleft. After that, we performed MD simulations to demonstrate that this binding is stable and also to identify critical acid residues which coordinate S3969 within this binding pocket. And these were phenylalanine 391 residue, tyrosine 406, and arginine 388. Particularly important was this interaction between phenylalanine and indole ring of S3969 because it was stable during the whole simulation. To prove this, we mutated these individual residues, and indeed, all mutations reduced or even abolished S3969 mediated ENaC stimulation. So our computer predictions was confirmed experimentally. And also in our MD simulations, we have noticed that S3969 binding to beta subunit causes a local conformational change. increases the distance between beta subunit and gamma subunit. So due to the binding, beta subunit moves away from gamma subunit. And this appears logical because the binding site is localized between two subunits at the interface between beta and gamma subunit. To confirm this, we again used our molecular leakage approach. We substitute two genes, one in beta subunit and other in gamma subunit by cysteines to form a new disulfide bond to be able to prevent this conformational change and also prevent channel activation by S969. And we were lucky to confirm these predictions experimentally. So this double mutant with the cysteine substitutions with this disulfide bond cannot be stimulated by S969 unless this disulfide bond is reduced by DTT. So on the basis of these results, we generated a simple mechanistic model of how S969 stimulates ENaC. So we propose that S969 binds to beta subunit at the interface between beta and gamma subunit, induces a local conformational change within beta subunit, which ultimately leads to increased open probability of the channel. So now we have characterized binding sites for inhibitory peptide and also for channel activator. And on the basis of this information, now as an outlook, we would like to identify novel ENaC modulators using virtual screening approach and automated patch clamp recordings. And we are happy to team up with Nanayan Technologies here in München to establish first ENaC recordings with the help of SynchroPatch 384. For these measurements, we obtained a stably transfected cell line, ENaC HEC cell line from Charles River, and we established a protocol to detect ENaC-mediated currents in this cell line. Also, we established a protocol to detect ENaC stimulation and ENaC inhibition. So we demonstrated applicability of this approach to detect both ENaC stimulation and ENaC inhibition. To stimulate ENaC, we again used S969 molecule. Here you can see an example trace demonstrating the recording from SynchroPatch. So the recording started in the absence of sodium in nominally sodium-free bath solution to avoid sodium overload of cells. After that, we increased bath concentration of sodium stepwise by changing the solution until concentration of 100 millimole was reached. And this high sodium concentration, ENaC-mediated currents can be detected. These currents can be stimulated by application of S969, which is reflected here by a negative deflection of the curve, and can be inhibited by application of amiloride at the end of the recording, which is seen here by a positive deflection of the current curve. In contrast, if we apply vehicle-controlled DMSO, no change can be observed, and application of inhibitory peptide inhibited the currents as expected. So all these effects were statistically significant, and we obtained pretty many recordings, successful recordings using this protocol, both for ENaC activation and ENaC inhibition. So importantly, success rate, so the probability to obtain a successful recording with ENaC currents was pretty high, about 70%, and therefore should be high enough to be able to perform screening experiments using this technique. So currently we are writing a manuscript to report methodological aspects regarding ENaC measurements in HEC cells using SynchroPatch 384. And we are also planning to establish a collaboration with the University of Zurich with David Penton to perform real screening experiments there. So at the end of my talk, I would like to thank all members of the Institute who contributed to this project, in particular Professor Christoph Korbmacher and my colleague Florian Suhr, who are here in the audience today. Also, again, I would like to thank Marcos Rapidius and the Nanayan team for helping me to establish these measurements of ENaC using SynchroPatch And also, I'd like to thank funding institution EFG for supporting this project. We are happy to contribute to Transregio 374 with this project. And of course, I thank you for your attention, and I'm happy to answer your questions.
Thank you, Alexander. That was a really awesome and concise overview of ENaC. I feel like I learned a lot myself. Hopefully the others too. Are there any questions from the crowd? Yeah.
I thought it was very, very interesting stuff. Do you have, is there any evidence for regulation of this cleavage of the inhibitory peptide? Is that regulated in different states? And why, I mean, in different states regarding hypertension, and so on? Or is that something that simply happens all the time, constitutive
process? We studied whether temperature 2 is regulated by aldosterone, and expression, function, and expression overall, and expression at the cell surface is not regulated by aldosterone. So temperature 2, proteolytic activity, and also ENaC cleavage is high, both without aldosterone and after aldosterone treatment. But aldosterone stimulates ENaC trafficking from ER through trans-Golgi to the plasma membrane. And during this trafficking process, aldosterone-mediated trafficking process, ENaC is cleaved. So indirectly, aldosterone treatment stimulates ENaC cleavage due to increased trafficking to the cell surface.
So it's not cleaved when it's in the apical membrane. It's cleaved before. Is that correct?
Probably ENaC is cleaved already intercellularly. Also, cleavage occurs at the cell surface to some extent. So in both compartments, this channel cleavage occurs. And what I wanted to say, when we stimulate the cells with aldosterone, we have much more channels expressed at the cell surface, but they are all cleaved, and this cleavage occurs during channel trafficking.
So you wouldn't be able to say something by measuring the peptides that appear in the urine at the end? Because, I mean, if they are cleaved on the cell surface, then they should appear later on.
Probably the concentration of these peptides
in a very low
too
concentration, yes, yes.
So we have never tried to analyze urine to try to find inhibitory peptides there. But probably this is an option, an interesting.
Question from Tom.
Thank you for the talk. I also learned a lot. And what struck me is that this is an open channel, basically, once it's cleaved. And this is an unusual situation, to my experience at least. Usually you have voltage gating or ligand gating or any other blocks like the Keir channel family. Is there any cellular component that stops the channel from being open, or is it not destructive to the cell's metabolism if it has an open sodium conducting pore?
So do you mean cellular compartments, intracellular organ?
Yeah, or with the Keirs, it is this polyamines that block it, for example, naturally at the resting membrane potential. Or no, it's when it's depolarized. But anyhow, is it really an open pore? And
so we and others studied ENaC function also in native tissues, and also in native tissues in lung and also in kidney. ENaC is constitutively active. It can be modulated. It can be modulated from intracellular side, probably by phosphorylation, or from extracellular side. But this is only a modulation of channel activity.
Okay.
ROMC is also open.
A comment?
Just a quick addition to your question. The main regulation is probably the number of channels expressed at the membrane. So insertion and retrieval, that's heavily regulated, and that determines the overall activity. Okay. Yeah, that's slow. That's slow because the kidney works slow. It's not like the brain. That's why we are kidney physiologists.